Hello everyone, today I'm going to be discussing an experiment in the Journal of Neuroscience in 2013 that talks about that's titled Physical Exercise Prevents Stress-Induced Activation of Granule Neurons and Enhances Local Inhibitory Mechanisms in the Dentate Gyrus. So granule neurons are essentially just the most abundant type of neuron in the brain, and so it's pretty much saying that exercise prevents stress-induced activation of those, so promoting a good kind of neurogenesis. And then there's also the interactions with the GABA neurotransmitter in the dentate gyrus. So we know that some background is that we know that exercise has something to do with us feeling good afterwards. It seems that exercise has some sort of protective effect on our brain's response to stress, and it promotes a more balanced neural activity. So now we're going to discuss that stress response and neurogenesis in the hippocampus. And this can obviously have some very crucial and direct effects for mental health research. So they essentially had two groups of mice. One were runners and one were non-runners. And they were adult male C57BL mice, and they were 6 weeks old. They were housed under controlled conditions, and they did neurogenesis labeling on them. So they used bromodioxyuridine, and they injected it intraperitoneally to label those new neurons. And it's able to incorporate once it recognizes DNA of dividing cells. So that's going to be that neurogenesis. And then the mice went under this cold water swim stress test. They're exposed to cold water for 5 minutes, 5 degrees Celsius. Both groups of mice were doing this, and that's supposed to be mimicking our natural stressors, because cold water activates this corticosterone in mice, which is sort of the cortisol for mice. And then they underwent some immunohistochemistry with some transcardial perfusion, so sort of preserving the tissue. There was anesthesia to make sure that they could have proper perfusion. And this essentially was recognizing the tissue distribution, because the immunohistochemistry visualizes that gene expression patterns. So then there was also microdialysis, which measures the GABA release during the stress exposure. So now we can see what inhibitory mechanisms are being used during that stress exposure. And then they were put in an elevated plus maze so that they could test stress in that way. And they used the elevated plus maze, and they just counted the entries and the time spent in the open arms. And then essentially, they did a craniotomy and a cannula implantation, where they infused the mice with bicuculin, which is able to block the GABA-A receptors. And they did this so that they can study the GABAergic inhibition role in the anxiety, so that now they can just see specifically GABA's role once it's being blocked, what happens. So this allows for direct drug delivery into the brain. Then they did their statistical analysis. They used t-tests and ANOVA to assess their different groups. And essentially they found that running increases the number of new neurons in the dentate gyrus, and it decreases anxiety-like behavior. So that's really important. Like I said earlier, these findings have serious implications for mental health research, therapeutic inventions. But there's still going to be further research needed to maybe do different types of stress tests, different types of animals, different places in the brain to look, maybe, and different ways that we can watch the inhibition. Running can essentially reorganize that hippocampus by influencing the GABAergic signaling, which affects new neuron production and then those neuronal integrations. So increased expression of the GABA transporter VGAT in runners which is what they found, suggests an enhanced inhibitory signaling, which may prevent stress-induced excitatory neuron activation. So yeah, running increases the number of new granule neurons but blocks the stress-induced activation of granule neurons. Yeah, so that's essentially what they found. Thank you.